and thank you for joining us for this next session, The Outcome of UKCTOCS. Where do we go now for ovarian cancer screening? We'd like to thank our sponsor, Merck, for making this session possible. The United Kingdom Collaborative Trial of Ovarian Cancer Screening, or UKCTOCS for short, was the largest clinical trial of its kind, and it provided critical data on the efficacy of currently available ovarian cancer screening strategies. Now, we are so lucky to have Dr. Usha Menon and Dr. Ian Jacobs, who led this study, with us here to tell us about the results of the study and what the future holds for ovarian cancer screening. Dr. Menon is Professor of Gynecologic Oncology at University College London in the UK, and Dr. Jacobs is President and Vice Chancellor and Professor of Women's Health at the University of New South Wales in Sydney, Australia. Also, don't miss the live Q&A they'll be doing this afternoon. They'll be zooming in from opposite sides of the globe to answer your questions. So thank you both for being here, Drs. Menon and Jacobs, and I will turn it over to you.
Good afternoon. It's a real pleasure to be speaking at the Ovarian Cancer National Conference. Thank you for the invitation. Professor Ian Jacobs and I will take you through our trial, the UK Collaborative Trial of Ovarian Cancer Screening. I will speak about how we organize the trial and the results, and Ian will follow me and tell you about what it means to the ovarian cancer community. These are my disclosures. And before we start talking about UKCTOCS, I want us all to think a moment about screening. So screening, as you know, is a way of identifying apparently healthy people who are at a higher risk of having a health condition. And the aim is to try and treat the individual earlier. And it's really helpful to think about screening as a sieve. So if you think about it, the screening test, if it's a sieve, we want to make the pores such that the cancer patients get through, and we hold back people who do not have cancer. And so as we increase the aperture, then we can get more and more cancer patients through, but at the same time, more people who do not have cancer get through. And so once the screening test, the initial screening test is done, we need to do further tests before we can be sure who might have a cancer. So before, the other thing I wanted to touch on, because, you know, we designed our trial in the late 90s and started it in 2001. So what we do need to do is take a moment to see, is it still relevant? Do we still need population screening for ovarian cancer? And for that, we need to look at the survival rates in ovarian cancer. And what I'm presenting is statistics that all of you are very familiar with. More than half of the women diagnosed with ovarian cancer do not survive 5 years. And this is the same on both sides of the Atlantic, and indeed across the globe. So it does seem that it would help for us to diagnose the disease earlier, so that the treatments, and now increasingly newer and better treatments, could work more effectively. And this is where we want to be with ovarian cancer screening. We want to have a population screening program similar to cervical and breast cancer screening, where women, large numbers of women come and get tested. There is also targeted screening, but that is not what UKCTOCS is about. Targeted screening is for high-risk groups, and you would be aware that in women with a family history, you know, there is targeted BRCA mutation testing, or smokers may have screening for lung cancer. But what UKCTOCS addresses is population screening, screening a large number of women with very few high-risk criteria for ovarian cancer. So just what does then population screening for ovarian cancer involved. So it's the same for all cancers. So it involves women regularly attending for screening, usually annually. It involves a huge health setup so that these women are invited, screening is done, and then when abnormalities or women at increased risk are detected, then they can have repeat tests. And then once the repeat tests are done, if there is persistent increased risk, then in the case of ovarian cancer, of course, the women will need to undergo surgery, and at the minimum, laparoscopic surgery, keyhole surgery to remove their tubes and ovaries, so that we can detect this woman who has ovarian cancer among all these women. So the questions that we explored are about understanding the risks and benefits of population screening for ovarian cancer. And in the trial, we call them outcomes. And our primary outcome, our main outcome, was to answer the question that in women who are invited for screening, will fewer die of ovarian cancer compared to those who have no screening? And then we had a number of other questions to understand risks and benefits. One is, would the screening detect more women at an earlier stage? The other is, will the screening detect cancers, good cancers that would not have been diagnosed during a woman's lifetime? You would have come across this concept in breast screening and in prostate screening. There has been quite a bit of debate in the public that sometimes the screening picks up cancers which grow very slowly and would never have been symptomatic or caused any problems during a woman's lifetime. The other questions are about harms, physical and psychological, and then whether it'll be cost effective, whether women will attend year for year, and how will the strategies perform. To answer these questions, we started in 2001, invited just over 1.2 million women. And by 2005, we had recruited over 202,000 women in the general population. So these were women who were aged 50 to 74. Their periods had stopped. As I mentioned, they had no high-risk family history. They may have had one relative with ovarian cancer, which was not considered part of the high-risk criteria. And then, of course, they should not have had previous cancer. We land ovarian cancer. We randomly allocated the women 50,000 to the blood group, 50,000 to the scan group, and just over 100,000 to the no screening group. The women had annual screens every year from when they joined the trial till December 2011. And we followed all the women using postal questionnaires, as well as linking them with their permission using the NHS number to the National Death and Cancer Registries, as well as the NHS Hospital Administrative Records. Our primary outcome, as I mentioned, is deaths due to ovarian cancer. And the date we chose to do this analysis was deaths up to 30th June 2020. Sorry, that should be 2020. So what happened in the blood group? In the blood group, women had a blood test taken, the bloods were transported overnight, and in our laboratory, we assayed the protein that you're very well aware of, CA125. We interpreted the CA125 value not using the usual cutoff or normal of 30, but using a special longitudinal algorithm, which we call the risk of ovarian cancer screening algorithm. There are other such algorithms, but the main key issue is that we were looking at a woman's CA125 values over time. And then depending on whether it had gone up or remained stable, we assigned risk, and women at low risk went for annual screening, and those with intermediate or increased risk had repeat tests. Over the course of the trial, just under 350,000 annual screens were performed, and women had an average of 8 years of screening The range was 7 to 11. In the scan group, women had an internal scan, a transvaginal scan, and we looked at their ovaries, and the ovaries were normally returned for an annual screen. Women whose ovaries could not be seen had a repeat scan, and where we saw an abnormality, they had a scan with a more senior person who looked and checked the ovaries and the tubes to see if it was suspicious of ovarian cancer. Here we performed just under 330 annual screens. And again, women had an average of 8 screens per on the whole during the course of the trial. So what were the results? I'm not going to go to the primary outcome now first, as is the norm when we report trials. I am going to take you through the story so that it makes sense. So it will be the secondary outcomes and leading to the primary at the end. So the first thing that we found was that women, 80% of women attended all their annual screening and repeat tests over the whole of the trial. So this is 11 years for some and 7 years for some. So all the appointments that we sent and any appointments for repeat tests, 80% of the women attended. And so that was in the blood group and about 78%, so similar in the scan group. So it's very clear that the women were very committed to the trial. And when we looked at the screening approaches and at their performance, in the blood group, 84%, so that would be 4 out of 5, just over 4 out of 5, of the women who developed cancer within 1 year of the screen were picked up by screening. The equivalent number was 72%, so a bit lower in the scan group. What I do want to highlight is in the blood group, half of the women we picked up had a CA1 to 5 at the annual screen less than 30, which is the usual cutoff. And we picked the women up because we were doing this longitudinal comparison, looking at their values over time. We could see the values were rising, even though they were within the normal range. Now, when we looked at women who did not have ovarian cancer over the course of the next year, did we call them low risk? Well, we got it right 99.8% of the time in the blood group and 99.5% of the time in the scan group. Of course, you might think, oh, these are really excellent numbers, but you need to think about it when these numbers in the context of the millions of women who would have screening if it was a program. So 99.8% means that out of 100,000 women, if we screened 100,000 women, 200 women we would call, would not have an ovarian cancer, but would be considered at increased risk and require repeat testing and go and have surgery. So that's the number. So 200 in 100,000, the blood group, 500, which is what 99.5% specificity is in the scan group. In the course of the trial, just over 2,000 women were diagnosed with ovarian cancer. And as you can see, almost equal, like over 1,000 in the blood and scan group together and over 1,000 in the no screening group. And when I say that they were diagnosed with ovarian cancer, this includes the women who had cancer detected by screening, the women we missed within 1 year of screening, as well as the women who never attended screening. They joined a trial, they didn't attend, women who had some screens and then did not return and then had ovarian cancer. So they had ovarian cancer more than 1 year after the last screen. And then women who were diagnosed with ovarian cancer after 2011, so between 2011 and 2020, after the end of screening. So we are looking at, and this is what we do in randomized control trials, looking at all women. So this is an intention to screen analysis. When we look at the women, and it's clear that there was no overdiagnosis. Ovarian cancer was diagnosed in about 1 in 100 women in all 3 groups. There was no difference. The blood and scan group did not pick up more cancers. And that is a good thing because we do not want the screening to pick up as cancers which do not have an impact on mortality. And these are just numbers, but if you did a formal statistical analysis, and this is the curves from the Lancet paper, you can see the 3 lines for the 3 groups, and this is the cumulative incidence of ovarian cancer. You can see the most important thing is that the lines all overlap, so there is no difference. Did we pick up more women with early stage cancer? And here, I'm just going away from ovarian cancer and focusing on invasive epithelial ovarian cancer. So I've left out the borderline tumors and looking at the invasive tumors. And ovarian cancer, of course, I mean tubal and the peritoneal cancers as well when I use that term. So we did this analysis twice, first in December 2014, 3 years after the end of screening, and then again in June 2020, more recently, 9.5 years after the end of screening. And you can see that in the blood group, compared, so just eyeballing the figures, in the blood group compared to the no screening group, we had about 11% more women diagnosed with stage 1 and stage 2. And then with the dilution and the cancers that have occurred after 2011, this has gone slightly down. So the difference is about 9%. But basically about 10% more women were diagnosed with in stage 1 and 2 in the blood group compared to the no screening group. But there was no difference between the no screening group and the scan group. So cancers were not detected earlier with the transvaginal ultrasound. Both the tests were associated with some harms, even if they were minor. So in the screening blood group, it was mainly bruising that women reported. And in the scan group, women complained both of pain and then some women complained of cystitis. And these were the rates. In 100,000 screens, we had a report of the test causing side effects in about 9 women in the blood group. And in 100,000 screens in the ultrasound group, 19 women. I'm sorry, I don't know what SAC is, but this is the scan group. 19 women, so more women in the scan group. But that was not the only thing. There were women, and you will remember from the diagram I showed you, there were women who underwent surgery and were found not to have ovarian cancer, but they had benign lesions or normal ovaries. So they had unnecessary surgery. And in the blood group, for each woman detected to have ovarian cancer, an additional 2 women had unnecessary surgery. If you look at it in another way, for every 10,000 women screened annually, 14 women had unnecessary surgery. And in these women, like in any other surgery, women undergoing any other surgery, there were complications, and it was 3 in 100 women had a complication. This number was much more in the scan group, where for every woman detected to have ovarian cancer, an additional 10 women had unnecessary surgery. And this comes to about 50 women for every 10,000 screens. While screening did not increase anxiety, there was some increased worry in the women who were found to have higher risk and were asked to have multiple repeat tests. On the whole, however, anxiety was not an issue. So having looked at all of these questions, we come to the primary outcome. Did screening reduce deaths from ovarian cancer? So during this trial, right up to the June last year, 1,206 women died of ovarian cancer. And as you can see, the number of women who died from ovarian cancer was similar in all 3 groups. This is not the exact way of how we do the statistical analysis, but I think it's an easy way to understand. So I've just put a simple percentage so that you can see that across the groups, it is the same. This is the way we analyze it statistically, the correct way. We look at the cumulative deaths from ovarian cancer from when the trial started, year 0 to 18 years, which is our end of follow-up in June last year. Again, you can see that all three lines representing the three groups overlap. Many of you would have seen treatment trials and see how survival curves separate. So these are all overlapping, and there is no difference in deaths due to ovarian cancer in either of the groups compared to no screening. And though there was a plan to analyze cost effectiveness, and we had done some preliminary work in this area back in 2015, as lives were not saved, we did not repeat this analysis recently. During the course of the trial, because of the generosity of the women, what we were able to build was a massive bioresource to support future research. We call this the UKCTOCS longitudinal women's cohort. So it contains a screening data from these 100,000 women and 650,000 animal screens, and then longitudinal follow-up data on the whole 200,000 women over time. We have over 500,000 samples and a very transparent governance and an access process so that researchers, whether from academia or industry, can access these samples. And these samples are unique because the women, firstly, these samples, most of them precede the diagnosis of an ovarian cancer and other cancers. So we have samples 1, 2, 3, 4 years before a woman was diagnosed with a cancer. And the unique aspect is in the blood group where the women gave a sample year on year, so that we have multiple samples, serial samples in these women, and they can be used to evaluate new tests to see if they would pick up the disease earlier. So in summary, though we provided the first definitive evidence that screening can definitely detect ovarian cancer earlier, and there was no life saved. So screening using, whether it was using the blood test or the transvaginal ultrasound scan, it did not reduce deaths due to ovarian cancer. And additionally, as you can see, it was associated with some harm. Some of the things we learned from the trial, which would be useful for others who are looking at other biomarkers, is that this longitudinal screening, that is comparing our women's samples to previous values, can definitely increase or improve the performance of screening tests. What these findings mean in the real world, Professor Ian Jacobs will tell you in a moment. But before I hand over to him, I just want to thank the women who took part, because without them, none of this would have been possible. And in 2012, we had an artist, Dr. Lizzie Byrne, paint a Graafian follicle, which is the follicle in the ovary, and digitize it so that each spot on that picture represents 8 women who took part in the trial. I want to thank the funders, the oversight committee, independent members, the trial team, and the senior investigators. Professor Jacobs will of course speak, and I want to name the others: Max Palmer, Steve Skates, Ali McGuire, Stuart Campbell, and Leslie Fallowfield. Thank you, and I hand over to Professor Jacobs.
Hello everyone, my name is Ian Jacobs, speaking to you from Sydney, Australia, and I'm delighted to follow on from my colleague, Professor Usha Menon, who will have given you an outstanding summary of the data in UKCTOCS. My task is to discuss some of the outcomes, give some interpretation, and leave you with some questions about where we go in the future. And I'm delighted to join this OCRA meeting. And I do bring you greetings from the beautiful city of Sydney in Australia, and from my university, UNSW, Sydney, the University of New South Wales, Sydney. Now, before I go any further, I do need to declare my interests. I was one of the co-inventors of the risk of ovarian cancer algorithm, which you'll have heard about. That has been licensed to Massachusetts General Hospital, and because of that, I'm entitled to a royalty stream through the company Abcodia, which now has a license to that algorithm. I also want to mention that, having read and heard about the work of OCRA, I was the founder of the Eve Appeal charity in the UK, which, like OCRA, is all about research funding, advocacy, and patient support for women who are unfortunate enough to experience gynecological cancer. And I am an emeritus trustee of the Eve Appeal. I am also on the board of Ovarian Cancer Australia, another wonderful organisation of a similar kind in Australia. And I send greetings from the members and trustees and supporters of those organisations. My involvement in ovarian cancer screening goes back a long way. I focused my research on trying to develop potential screening tests for ovarian cancer back in 1985, when I was a surgeon training in gynecological cancer, and was frustrated by the poor outcomes for the women we treated. And I felt that screening and early detection was the way ahead. Soon after that, I was joined by Usha Menon and other brilliant researchers, and this has led to a program of research over 35 years, which has culminated in UKCTOCS. And there are lots and lots of aspects of this extraordinary research program, which has involved, amongst other things, 200,000 women in the UK. Lots of aspects of it that we don't have time to talk about, but you've heard from Usha about the data most recently published in May 2021 in The Lancet. And I'll start with some conclusions to follow on from Usha's description of the findings. The first few conclusions are very positive. It is possible to detect ovarian cancer early by screening. That's wonderful. 30 years ago, people really didn't know if that was feasible. It is possible to do it using the risk of ovarian cancer algorithm, followed by ultrasound and by ultrasound. Even better than that, it's possible to screen many, many thousands and thousands of women repeatedly over many years with high acceptability and a very, very low complication rate. And the third piece of good news is that the multimodal approach to screening, that's using CA125 in the risk of ovarian cancer algorithm followed by ultrasound, can achieve a shift in stage. And by that I mean that more women are diagnosed as a result of screening with early stage disease and fewer with late stage cancer. Now that, of course, all sounds wonderful. If you can pick it up, it's highly acceptable, the complication rate is low, and you can shift the stage, you'd expect that to solve the problem. But unfortunately, that shift in stage and the early detection did not result in a reduction in the number of deaths from ovarian cancer in the UKCTOCS trial. I'll repeat that again. Although we can pick up the cancers, over 85% of women with ovarian cancer were picked up early by screening. Although it's highly acceptable and women came back year after year and the complication rate was low, and although we achieved a shift in stage to more women with stage 1 and 2 cancer and less with stage 3 and 4, despite all of that, the stage shift did not result in a reduction in the number of deaths. That's deeply disappointing. desperately disappointing for everyone involved and frustrating that we're unable to do what we've sought to achieve over 35 years of research, and that is to save the lives of the women threatened by cancer of the ovary. And you can see here some of the headlines when that Lancet paper came out. In the UK, in BBC News, setback as major screening trial fails to save lives. And in the Sydney Morning Herald, where I am now, desperately disappointing setback for ovarian cancer screening hopes. It really is a devastating finding. But we have to face up to that and take on board the key message that results from that. And that is that for now, we cannot recommend general population screening for ovarian cancer as a national program. And the reasons are, of course, that a screening program amongst a whole population does cause some harm in terms of anxiety and in terms of false positive results that lead to surgery, and it has a major financial cost, and that cannot be justified without evidence, definitive evidence, that screening saves lives. So for now, we cannot recommend population screening for ovarian cancer. And that leads on to a whole raft of questions, discussion points, and issues that I hope, once I finish my presentation, we'll have time to discuss when Usha and I join you for a question and answer session. And I've lined up 7 discussion points. You may think of others. The first one, what does this mean about the length of early detection, the time period before the cancers would otherwise have been diagnosed clinically, that is needed to achieve early detection, which can save lives? We think that in UKCTOCS, the average time before clinical diagnosis for the women detected by screening was about 18 months. And clearly that was not enough early detection to save lives. How far forward would we have to go with early detection in order to make screening effective? Would it be 3 years, 5 years, or 10 years? I don't know the answer to that, but those questions face researchers who are now trying to develop the next generation of screening tests. And of course, we do have the resource for them to be able to ask those questions, because we do have the biobank, the samples collected from the women who participated in UKCTOCS. We have that biobank for them to work on to see how much earlier, using those samples and new tests, ovarian cancer could be detected. It may be that you need to pick up the cancer as long as 5 or more years before clinical diagnosis would have occurred in order to effectively save lives. My second discussion point is around the question, are there potential tests in development which can improve or may improve early detection? And the answer to that is encouraging. There is incredibly powerful proteomic technology, genomic technology, and other technologies which are being studied right now, which may well prove to be effective in early detection of ovarian cancer and may well save lives. The rate of development of science and technology, some of it I'm sure supported by OCRA, is phenomenal. So although the results of UKCTOCS are deeply disappointing, we can still have hope for the future that better tests will either add to what CA125 in the risk of ovarian cancer algorithm can achieve, or independently achieve what it couldn't achieve. So I remain optimistic, and there is plenty of room for optimism. And that leads on to the third discussion point. If a new test is discovered, which seems to detect ovarian cancer very early, perhaps 5 or 10 years before clinical diagnosis. How long would it take to prove that it works? And this is another challenging message, because a lesson from UKCTOCS is that we can't rely on measures like stage shift to predict that a screening test will save lives. We have to do another big randomized controlled trial like UKCTOCS. And that trial, as you've seen, takes a very long time to do. It will probably take 5 to 10 years before it is feasible to justify doing another trial, and then another 10 to 20 years to complete the trial. So although I'm very optimistic that there will be better screening tests for ovarian cancer, which can be evaluated in a big trial, it will take time to do that. Because of that, I would predict that general population screening for ovarian cancer, although I'm confident it will happen eventually, I would predict that it is over 20 years away from now. That is a difficult message for all of us to take, but I think that's the truth. Now, given that we're unlikely to have screening for ovarian cancer in the general population for quite a few years, is there any other role for screening? Could screening for ovarian cancer be used using the technology that we have, for instance, amongst women who are at very high risk of ovarian cancer, those who have a very strong family history or a genetic predisposition due to a mutation in a gene like BRCA1 or BRCA2? Now, this is a question that will now be debated given that the UKCTOCS results are out. There is separate data from non-randomized screening trials amongst the high-risk group of women from 2 studies in the USA and a big study, UKCTOCS, in the UK, which shows that amongst women at high risk, screening does pick up almost all the cancers early, that it is able to shift stage to earlier stage, and that it makes the surgical treatment of the cancers less complicated. That is all encouraging, but of course there is no mortality data. There is no data from those trials to show that it reduces the number of deaths. For women at high risk at present, the options are essentially do nothing or have surgery to remove the ovaries and or tubes. So it is possible, but this is a controversial topic which will need more discussion and debate. It is possible that an interim option for women who, after counselling, have decided that they want to delay surgery or that they don't want to have preventative surgery would be to have screening using the risk of ovarian cancer algorithm with ultrasound for a period of time. I'm sure that'll come up in our discussions in a moment. My 5th point relates to the implications for symptom awareness campaigns. Many organizations that care passionately about women faced with ovarian cancer have advocated for symptom awareness. And it is, of course, really important that women do not ignore symptoms that may be ovarian cancer. But I think we have to be honest with women. Of course, act on symptoms, but we should not hold out to them that that will improve the outcome for ovarian cancer if they have it. The women in UKCTOCS were detected before they had symptoms, and we were still unable to save lives. So it is extremely unlikely that knowing that women have symptoms earlier will result in saving lives, albeit that there is very good evidence that it will improve the way they are looked after, the ability to send them to the right place for their care, and it may well be a benefit to them in terms of the anxiety and other factors around ovarian cancer to have the disease identified early so that they get treatment early. But I think we have to be honest and say there is no evidence that symptom awareness will save lives in ovarian cancer. I have 2 more points. My sixth point is to ask the question, what does the lack of improvement in survival for women whose cancers were detected early tell us about ovarian cancer? Or perhaps say, why did those women not have a better outcome despite the fact that they were diagnosed at stage 1 or 2 instead of stage 3 or 4? What seems to have happened in UKCTOCS is that we picked up cancers that had a poor outlook. Instead of them waiting to be clinically diagnosed at stage 3 or 4, we picked them up at stage 1 or 2, but they still behaved as they would have done if they hadn't been diagnosed till stage 3 or 4. That's deeply depressing and frustrating, but it probably tells us quite a lot about the underlying biology and pathology of ovarian cancer. And there's a lot of work that can be done now using the UKCTOCS database, the samples from UKCTOCS, to try and understand that better, which may be really important in understanding ovarian cancer and in developing better methods of early detection and screening. My seventh follows on from that. If we understand ovarian cancer better, it may be possible to develop new treatments for the women with ovarian cancer who are detected early by screening. So I have a little bit of hope that detecting cancer early in women in that category may lead us to new therapies and may make that early detection worthwhile. At present, the treatment that women who are detected early and have stage 1 and 2 cancer, the treatment they receive is the same as women who are detected by symptoms at stage 1 or 2 receive. Perhaps women who are detected early by screening need a different type of treatment. That is another, I believe, important area for research, which may hold out some hope for the future. My last discussion point is to just point out to all of you that there are lots and lots of other benefits and lessons from UKCTOCS. This massive trial and the experience from it will guide the design, the analysis, and the approach to future large-scale trials in ovarian cancer, in other cancers, and in other diseases. There are an enormous number of lessons there to be learned. There's a huge set of data about ovarian cancer and other cancers and other diseases for researchers to work on. One of those is the biobank of samples from trial participants, which is right now being used for research and discovery of new biomarkers. And I talked about the hope that we can find new and better biomarkers. And those biomarkers are, of course, in ovarian cancer, but the biobank is applicable to other cancers and other diseases. And that biobank is now available to be shared with researchers across the world. 3 other quick points. I've already talked a little bit about the insights into the natural history of ovarian cancer that the UKCTOCS experience could provide. UKCTOCS has trained many truly outstanding researchers and scientists over the years of the trial. And there's one other real benefit. We have a unique quality of life data set, serial data on quality of life collected from 185,000 women who took part in that trial, which is unique and extremely valuable. That's the story. Those are the discussion points and implications. I want to finish by acknowledging, first of all, and I know Usha will have done this, but it's important that I do as well. I want to acknowledge the funding agencies. Putting together a trial like this would be difficult anywhere. I think it would be almost impossible in any other country in the 90s and the 2000s to do it. It required the Medical Research Council, the National Institute of Health Research, the NHS R&D funding agency, Cancer Research UK, and the Eve Appeal to come together in an extraordinary way, and I will always be grateful for that. It also required an amazing team of investigators. You've heard from Usha Menon, Stephen Skates, Max Palmer, Leslie Fallowfield, Stuart Campbell, Ali Maguire, other co-investigators, a wonderful coordinating team at UCL. You see a picture of us there when we were a bit younger. A rigorous independent oversight committee, a data monitoring and ethics committee, a trial steering committee. These people put in hours and hours of work. Other collaborators in UKCTOCS, UKFOGS, and internationally, and some of those are, of course, great researchers in the USA that OCRA will be familiar with. And of course, last but definitely not least, the 202,000 women who gave their time and their dedication to participating in this trial over so many years. I am deeply grateful to them. Finally, I just want to finish by saying that the UKCTOCS team, Usha, myself, and our colleagues are desperately sad that our 35-year quest to save the lives of women who develop ovarian cancer has not succeeded so far. We'll continue with our determined efforts. Nevertheless, we're proud of the way this complex study was conducted. The trial did answer the question it set out to answer. Could screening reduce mortality? Sadly, that was negative. It's contributed to understanding of ovarian cancer and to the design of future trials. And I reflect that research, unfortunately, doesn't always go in the direction you want. It doesn't always go in straight lines. Often the results are surprising, or as in this case, disappointing. But if research is properly conducted and rigorously conducted and honestly conducted, it is always worthwhile, and it always lays the foundation for future progress. And I'm confident that that's what we'll see. I'm confident that in due course, we will have an effective screening test which will save the lives of women with ovarian cancer. And I know that that's what everyone at the OCRA meeting and everyone involved with the OCRA desperately wants. Thanks so much for giving me an opportunity to speak to you, and greetings from Sydney and Australia and from the University of New South Wales in Sydney. Thank you.